안녕하세요.
Welcome. Okay, let's start the count quiz first. Now this should be straightforward. Okay, first generation cancer drug is blocking the fast cell growth, right? So what is the target of the capsule?
Microtube.
Okay, can you give another example of the first generation of a cancer drug?
The DNA replication blocker or
For example?
topoisomerase blocker.
Any specific compound name? Most famous one is cisplatin. The compound is still #1 drug when you have a cancer, and that is the first choice so far. It's a DNA attacking compound. So those kind of DNA attacking or microtubule attacking compounds, those are the first generation. Okay, so specifically activated pathways or the target enzymes or receptors can be second generation of the cancer drugs. What is the other name of a second generation of a cancer drug?
Targeted cancer therapy.
Targeted cancer therapy, right? Targeted one. Cell cycle checkpoint is also targeted cancer therapy. They can be kinase of a cell cycle, the controlling. Do you remember the name of those kinase? Kinases in CDK, cyclin dependent kinase. Those kinases are also target for the targeted cancer therapy. So PD-1 is the third generation, which is the switch off for the immune system. If you block it, then we stimulate the immune system. NK cell is also kind of a lymphocyte, even though the cells are not involved in adaptive the immune system. But NK is also close to B cells and T cells. Macrophage is a typical myelocyte cells. Okay. Okay. In CAR T, chimeric antigen receptor T cells, the changed part is a TCR. T cell receptor was changed with what?
SCFB.
SCFB.
Usually that the SCF or the FB was replacing the TCR. Okay. Originally, the position was the TCR. If TCR does not work properly, then we introduce different binding motif, antibody mimicking SCF. That's the CAR T concept. Okay, so this is all we have today. So for today, I invited the presenters. Yonghun is going to show you one kind of a PD-1, the blocker. So there are two kinds in the market right now. So two of you chose the other one. Yonghun chose these cases. Now this case is specifically interesting because this drug has been applied to a famous person. It's the US President, Jimmy Carter, was diagnosed as the cancer, and his expectation date was not so long, but by treatment of this drug, he was fully cured. So that's why it became quite famous. So this gives you some hint that if you choose the interesting story, okay, science part should be exciting. That's most important. But if there is any other stories you can create together, then it would be easier for you to attract the attention of those audience. Okay, so Yongun will be the first presenter today. And Tekjun chose the different antibody and targeting different one for 1BB, not the PD-1, not CTLC. So this one will give you another example of the idea of the antibody treatment as immunotherapy. And then Dong will talk a little more about the CAR-T. So with this, hopefully we can learn almost everything about this third generation of cancer. Okay. So Yongwon, would you start?
Okay, let me start the presentation. Pembrolizumab, also known as Keytruda, is the drug that I will talk about today. Firstly, I will give you the brief background information about this drug, and then I will talk about the mechanism and the adverse effects that is caused by the mechanism, and some cost and effectiveness issues about this drug. And then, although those costive and effectiveness issues, the significance of third generation drugs. Firstly, as Professor said, you may know this guy or not. This man is the Jimmy Carter, who was a former president of the United States. He was prescribed as in the termination stage of the metastatic melanoma at the age of close to 100 maybe. But he was little bit 겸허하다, 약간 little bit, and
modest or humble.
wise or humble about his state, but he was alive by the miracle. He was injected Keytruda or pembrolizumab, and then he was perfectly cured by this drug. Even though his melanoma cancer was metastatic, so this cancer was spread to even to his brain, but he got perfectly cured. This drug was invented by the pharmaceutical company named MSD, and as I said earlier, It can be utilized to metastatic melanoma, metastatic non-small cell lung cancer, and metastatic bladder cancer, and etc. Uh, this drug looks like this picture, and it is in the class of IgG, and specifically it is in the IgG4. It is made from inside of the mouse, and scientists humanized it to be used in the human's body. Mechanism is this targeted antibody, when they are in the human's body, they bind to the PD-1, which is on the T cells surface, and then so PD-L1 or PD-L2, which is on the APC or cancer cell, cannot bind to the PD-1 of the which is on the T cell's surface. So T cell can kill the tumor cell or APC. But by the bind of the PD-1 and pembrolizumab Keytruda, this can also cause adverse effects, because the boost of immune system means that it can kill another normal cell, because on the surface of the normal cell there are also the P-L1 or P-L2, because cancer mimicking normal cells method. So if pembrolizumab binds to the surface or binds to PD-1 on the T cell, then P-L1 or P-L2 of the normal cell can't bind to the T cells P-1. So boosted immune system can kill normal cells very much. So as for the adverse effects for that, inflammation can happen on many sites, such as lung, thyroid, pancreas, and colon and liver. And also there are some cost and effectiveness issues. Firstly, this third generation drug, including Keytruda, are very expensive. For Keytruda, it is administered via injection, and for one injection, it costs about $5,000. And also there is maybe more expensive one, this one, in the third generation drug. And it is also reported that most immune checkpoint inhibitors, including pembrolizumab, is effective for 20% patients who are prescribed. So the case that I introduced earlier, the Jimmy Carter case, is the very exceptional case. Then it has cost issues and effectiveness issues, but immune checkpoint inhibitors are definitely the third generation drug of the cancer therapy, and it is very researched by a lot of people. Then why is it? Because this therapy may be the essential way to get rid of the cancer. Essence of immune checkpoint inhibitors is the activation of immune system. The classical drug usually targets cancer itself, targets cancer, and classical drug or surgery is the solution from outside and targets cancer itself. Cancer drug targets cancer, but this immune system is the solution that is given inside our body. And so some cancer cells are originally mutated and change themselves fast. So as I said earlier, drug or surgery may not be effective, and also these solutions are from the outside of human's body. So some sites of cancer may be hard to be removed or hard to be removed by surgery, also hard for drug to reach. But immune system can cope with these situations well. Firstly, it can cope with mutations well, and the immune system is in our body, so it is easier for the immune system to approach to the anywhere where there is cancer. Uh, by these things, immune checkpoint inhibitors are significant, and many scientists are trying to develop such drugs. Thank you for listening to my presentation.
Okay, thank you, Young-won. Now, could you give questions to Young-won?
I have a question.
Go ahead.
So when you treat the Keytruda to any cancer patient, can synergy be expected when used with CTLA4 blocker together? Or is there no connection?
I cannot remember exactly the synergy with the synergy can happen with CTLA4, but I read the book, The Understanding of Bioscience, and on now they for the Keytruda or other third generation drug is now developed to be administered by other drugs with other drugs that as I remember, and and so I also think that it can be there can be synergy with the drug that inhibit the CTLA4. As I know.
Young-won, are you saying that the administration method is different or material is different?
I mean two different materials is administered 병용투여. I mean.
Okay, so material is different.
Yeah.
So your accent was on administered. So that gives some impression that maybe the administration method is different or material is different. So your focus is on different material. So
Yes.
two different drugs can work together. Then you can call as a 병용, it's combined, or you can call the cocktail approach. So you mix them together, and in principle, if their target is different, yes, it's possible to use the both of them at the same time. But if their side effect is also related, then it can be a little more dangerous. Side effect also can be increased. Other question?
I have a question as well.
Yeah, go ahead.
So in the slide where it says that it only had an effect of 20%, I think it was like the last slide.
Can you say it again?
Oh, okay. So I can't understand what the 20% actually mean, because the patients could be either in the last stage of cancer or the early stage of cancer, and those efficiency can be different in those all kinds of patients. So does that 20% mean that it has an effect of the total patients that have been all prescribed after this drug has been approved by the FDA, or is it only 20% or on certain standards?
What is the meaning of the 20%?
Maybe it is written little. It is written, as I know, not much in detail. So I think that this means after the approval of FDA, and then to the patient who is administered this drug, I think maybe all the patients who is administered this drug.
Okay. if this 20% is covering all stage of patients, early stage or later stage, why 20%, you think? Why only 20%?
I think because of the too hyperactivation of the immune system may be the reason. To properly regulate the immune system is not that easy or for now circumstances, I think.
Well, maybe those are side effects, maybe one possible reason. But if they analyzed the further, maybe the mechanism, if those are patients, the cancer cell, if they do not express PD-L1, then there is no target available on cancers, then maybe the drug may not work. So that correlation is not clear for now, I guess. But if they can check those expression of PD-L1, then before they start this treatment, they can decide whether this patient can be susceptible or effective for the drug or not. Okay. So similar things. Go ahead.
You mean there are some patients whose T cells does not have PD-1?
PD-L-1.
PCR.
So if you can check whether the patient's the cancer has that target or not, that can be good guideline whether this drug may work or not. Right? So that kind of a pre-checking is also important. Same thing for the targeted cancer therapy. If that patient does not have that tyrosine kinase overexpressed or mutated, then there is no point to use those drugs. So that kind of person-specific the treatment is more and more getting popular. But so far, we don't have enough options of drugs. So that's why we needed to collect more specific drugs that we can choose. In the middle of the Young's the presentation, Young said maybe more than 10 times of dash, PD-1. What is a dash?
I'm sorry.
No. That's everybody, all of us use that word. 1-3, 1-5, we use that. Where is it from, do you think? Is that from, is it from Japan? Then what is the origin of the word, do you think?
Dash.
It can be dash, D-A-S-H. It's a dash. That English word, maybe somebody pronounced maybe by Japanese, and we just say dash, but maybe the origin is the English word dash. Okay, this the antibody is a humanized antibody, you said. That means this is originally developed in mouse or rat. Why do we need to make a humanized? Why don't you just use a human antibody? Then we don't need to make a humanized, right?
Because we can use a human as a experiment object.
Yeah, true. So we cannot inject the vaccines.
Also, mass production is a lot harder for human when we use human as a antibody source, maybe.
What do you say? What is harder?
Mass production.
Mass production. Hey, mass production is anyway, you don't use mouse, right?
Yes.
You get mouse antibody, then you replace majority of the part with the human sequence. Anyway, the synthesis, the production is done in some cells. What is the name of the cell? Anybody?
Hybridoma cell.
Hybridoma cells. Hybridoma cells is a hybridoma or chimera. That hybridoma cell is composed. It is made by which cells?
Myeloma.
B cell and myeloma cell.
B cell and myeloma cells. Myeloma is a cancer. Okay, and then they produce this the specific monoclonal antibody, and later you needed to modify the majority of the part by replacing of those sequence with the human sequence too. So that's a lot of work in there. Okay, as a side effect, Yongwon said there may be some inflammation. Then high inflammation is it good or bad for cancer treatment? What do you think? If we wanted to attack cancer, we want to increase or decrease inflammation.
Want to increase inflammation.
So very briefly speaking, to attack cancer, we needed to increase inflammation. If we go to too high, then too high inflammation is toxic to us. So this is the balance in between inflammation and cancer. Okay. So if you get low inflammation, we have a better chance to have a cancer. It's a paradoxical, but that's kind of the, the rough idea, the in between inflammation and cancers. Okay. So let's just stop here and let's move to the questions. Thank you, everyone.
Yes.
Okay, question.
Okay. I will introduce about urelumab and
Actually, would you
utomilumab.
speak a little louder? Your voice is a little too small.
Okay. I will introduce about urelumab and utomilumab, and these drugs is briefly introduced in bioscience 이해. And these two drugs both targeting 4-1BB. The other name is CD137, and this is a monoclonal antibody, specifically agonist antibody. And this kind of agonist antibody makes immune system stronger. We in the video clip, agonist antibody introduced very briefly, and both urelumab and utomilumab drugs aren't complete yet, but still being in clinical trials. The strategy of targeting 4-1BB is quite different with anti-PD-1 or anti-CTLA-4. In case of targeting PD-1 or CTLA-4, the functions of PD-1 or CTLA-4 is applying the brakes on immune system. So when we use targeting PD-1 or targeting CTLA-4 drugs, we can remove that brakes so the immune system can do their works well. That is the strategy of anti-PD-1 and anti-CTLA-4 drugs. But anti-4-1BB drugs, these drugs leads to pressing the accelerator of the immune system. So when we use these anti-4-1BB drugs, we can make immune system stronger. So this is quite different strategy. 4-1BB protein is expressed on T cells and natural killer cells and innate immune cells. And these 4-1BB proteins binds to 4-1BBL, 4-1BB ligand, and this 4-1BB ligand expressed by antigen presenting cells and dendritic cells and macrophages. So when we use anti-4-1BB drugs, we can activate the 4-1BB proteins and we can make immune system more stronger. So what happens when 4-1BB proteins activate? In case of T cells and natural killer cells, they upregulate the anti-apoptotic materials, so they can live longer than usual. And also they induces proliferation, so increases the number of cells of T cells and natural killer cells, so they can do their works more well. and they also produces interferon gamma and interleukin 2. When interferon gamma produces from T cells and natural killer cells, interferon gamma recruits other immune cells, and also interferon gamma stimulates to produce interleukin 12, and that interleukin 12 is produced from dendritic cells and macrophages, and via positive feedback, interleukin 12 also stimulates to produce this interferon gamma. So this through this positive feedback, the immune system gets stronger. And when interleukin 2 produces, interleukin 2 promotes the differentiation of T cells to become memory T cells. And the other one is increases the secretion of perforin and coenzyme on both T cells and NK cells. We have already seen the video clip that perforin and coenzyme is comes from T cells and NK cells and attack the cancer cells. So perforin and increases the secretion of perforin and coenzyme makes their works more stronger. And this is the mechanism. In recent clinical tests, both urelumab and utomilumab used with other drugs, such as anti-PD-1 and anti-EGFR monoclonal antibodies, to check the efficacy. And not only in urelumab and utomilumab, other anti-4-1BB drugs are also being used with other drugs, such as anti-CTLA-4 and anti-PD-1 antibodies, to check their efficacy. This anti-4-1BB drugs topic is very new topic, so many experiments and tests are ongoing. Thank you for your listening.
Okay, thank you, Taekjun. Now, guys, the question to Taekjun. Taekjun said this antibody is an agonist. If this is agonist, then how do you define the PD-1 anti-PD-1? Would you define what is an agonist?
Agonist, the word. Means makes more stronger.
Agonist makes the receptors, the original functions stronger. So that's agonist. What is the opposite word of agonist? Nobody knows?
Antagonist.
Antagonist.
Antagonist, okay? You can put A and T, antagonist. Then the question is, if this antibody is an agonist, then how about the PD-1 antibody? PD-1 antibody, should you call that as an agonist or the antagonist?
Antagonist.
Is it antagonist? So by adding PD-1 antibody, you can boost the immune systems. You can make a stronger the immune system. Then is that antagonist or agonist? What do you think?
Antagonist.
Can you explain why?
The function of PD-1 is breaking the immune response by binding PD-L1. So, which means blocking PD-L1 makes the immune response stronger it is antagonist.
Yep, that's the right way we can see. So original function of a PD-1, if it is binding to its agonist, the PDL1, that's agonist, then their signal is suppressing the immune system. That's their original function. So that's why PDL1 should be considered as an agonist here. Then if you add the antibody, either binding to the PDL1 or PD-1, they are competitive inhibitor. And if they block the function of original PDL1, you should call that as the antagonist of a PDL1. Okay. But in this case, interestingly, 4BB or the 4BB, in this case, antibody itself binds to the receptor, then it activates. It's also competitive, kind of a competitive agonist, you can call, by binding there, and then you can actively stimulate that receptor's function. Sometimes that is possible. That means PD-1 too, if you use a PD-1 antibody, there is a chance that this PD-1 antibody it can activate the PD-1 function. That's also possible. But the currently used the PD-1 antibody that act as antagonist, blocking, and it does not induce the function. Okay, so that means if you have a receptor, target receptor, depending on those binding mode or the structure, some molecules they can act as an agonist, and sometimes it acts as an antagonist or inhibitor. Okay, so that's case by case. Any other questions, protection? Both Youngwon and then Taekjun mentioned that the expression can be either on tumor cell or it can be on APC, antigen presenting cell. If their target is expressed on an antigen presenting cell, if this T cell is activated, then APC is safe. APC is not cancer cells. APC is kind of part of our immune system. If they have the same target, if they activate the T cells, then they are not in danger. What do you think? The T cell attacks the tumor. Yeah, go ahead.
I think they'll be kind of different in somehow.
Could you say a little louder?
I think they could be a little bit different from other toxic cells. They have other receptors. Maybe.
Yeah, it's not clear, but there may be some other defense mechanism for APC. They don't want us to get attacked by T cell through perforin or the granzyme, and they will die right away. APC's role is that they need to secrete other signaling molecule to attract more the immune cells from others. So even though we do not know that exact the mechanism, but we can assume that there is maybe different defense mechanism. Don't let the T cell attack directly to the APC. That's just something we can imagine, right? So now, if you imagine that kind of a possibility, that we call this possibility as a hypothesis, 가설. So if you set up this kind of a hypothesis, then your research can be, okay, I wanted to find out the defense mechanism of APC from the attack of T cells, something like that. Okay. Immune system is quite complicated. There are T cells, B cells, and there are helpers, and also antigen presenting cells here and there. And in general, we said increased immune system, increased inflammation may help to remove enemy or the foreign bodies. If it goes too high, it's also dangerous. Then we need to get balance. And which one we should kill, which one we should save. So everything is quite complicated. But in general, those are the cells working in high inflammation area. Anyway, they are too much activated, and anyway, their lifetime is usually very short. Even the T cells, APCs, they don't live long. That's why there's a lot of bodies accumulated. So that's kind of, from the wound, we generate a lot of the garbage materials. Okay, if you don't have more question to Taekjun, then thank you, Taekjun. Let's move on to third speaker, Dongwoo.
Okay, so I'll start my presentation.
Could you speak a little louder?
Okay, can you hear me well?
A little closer to mic, still a little small.
Okay, I'll increase my volume, so, like this.
Better?
Okay. So I'll talk about CAR T today, and as I said in the title, a brief history of Kimra, which is the first drug which have been developed by the CAR T technology. So immunotherapies are third generation drugs to treat cancer, and they have been coming out in the market since 2010. So you can see that various types of immunotherapies, including vaccines and checkpoint inhibitors and other very diverse things have been coming out in the past 10 years. And CAR T has been even a groundbreaking technology, even in the developing stages of immunotherapy. So there are only two drugs, according to the 2017 statistics. So it is a new form of immunotherapy. And we have talked about the various mechanisms of it. So first, you could see that the receptor, the TCR receptor of the T cell in the normal T cell usually binds with the MHC1 class 1 of the tumor cell, which present an antigen. However, this TCR is modified, and the ectodomain, the domain that is facing out of the cell's membrane, is changed by a single chain factor, the SCFV. So this actual abbreviation is, I think, single chain factor, variable factor. I don't know the
Fragment
exact
variable.
abbreviation.
Fragment.
Okay. Fragment variable. So, sorry. So this means that it mimics the structure of an antibody so that it could bind to a specific target in the tumor cell. So you can see that the CAR T cell is generated and it has an antigen recognition domain, which is over here, and also a signaling domain that relates its signals to the T cell to accelerate its attack. So this process is usually maintained by injecting a viral vector to the T cell, which incorporates the genes inside to the T cell's nucleus so that it could express proteins that could have a newly generated chimeric antigen receptor. So this is the exact mechanism of how the viral vector is transfected is currently, I think, a secret of its companies. So I didn't have the permission or like the exact mechanism to know how it exactly injects, but this is the general impression. So after the T cell is collected by the patient, it gets binds and fused into the cell and gets integrated, and it is again then transferred again to the patient. So this is kind of an autologous therapy, which is to bring out the T cells of the patient's own and to inject it again after it gets modified. So after then, the patient gets constantly monitored with disease responses, and it has a persistence of the CAR T cell. So I will also talk about the brief history of the first CAR T drug, Kimra, because I thought it was an interesting topic to tell you about. It has very interesting features about how different companies achieved and this didn't achieve. So the first discovery of this CAR T was made by the professor of University of Pennsylvania, Carl June, and this professor have found a new type. Oh, I didn't explain this. So the CAR T has first generation and second generation and third generation. So after this generation is being developed, many factors have been added to make it more efficient. And the first CAR T cell, Kimra, was actually a second generation CAR T cell. So this targets the CD19 antigen in tumor cells. And as this professor names it, he tells the media that these modified T cells are serial killers because the T cells can, one single T cell that has been modified can destroy up to 100,000 cancer cells. So it's a turbocharged T cell. And this is actually a Google map. And this shows that the first company to develop this drug after getting the idea, which was developed by Carl June, was Novartis. So this company, Novartis, is in the top of the Charles River where the MIT is. It's very close. They're very closely located to each other. And so this is the place where CAR T has been developed. So I also wanted to mention there that Harvard is down here. And over here, there's Merck, the company Merck, which is also a pharmaceutical giant. So in here, they develop PD-L1 drugs, which Young-Hoon had talked about earlier. So the Novartis have made a clinical trial in 63 patients who were suffering from leukemia, and this disease was very severe that it couldn't be cured by many other previous cures, and they are basically ready for it to die. So they got this new clinical trial, and of the 63 patients, 52 of them survived, and 11 of them were unfortunately dead. But still, this means that this CAR T cell was a groundbreaking technology. So the most of the death was occurred by this side effects called one of the two, B-cell aplasia or CRS, or cytokine release syndrome. So these two things are caused when the immune system is over-exaggerated, and they also target normal cells, not only tumor cells that were expressing CD19. So Novartis had a way to overcome this, had developed a method to overcome this side effect, which is to remove the complete immune system of the patient and let them to recover in a septic room, which is 무균실. However, another company called Juno wanted to also develop a clinical trial based on Carl June's technology, but eventually failed because too many patients have died out, while they hadn't had a chance to overcome the side effect. So this Kimra is designed for ALL, which is acute lymphoblastic leukemia, patients who are very young, and also for older patients who are suffering from similar diseases called DCBCL. So this is basically all diseases that cause from the impaired function of the B cell. So Kimra generates destroys not only leukemic B cells but also healthy ones. So the patients need treatment to protect them from infection. So after they get this dose of Kimra, they receive infusions of immunoglobulins every few months to overcome this side effect. And the second thing of this chemera is that they cost about 5억 won, so it's very expensive. Even though it's refundable, is that if the treatment is not working, it's still very expensive. So many patients are reluctant to get treated by this disease. But nonetheless, it's very effective if you get treated. So this is my last slide, and I want to talk about how the future of CAR T cells could be. And the picture over here is actually a 12-year patient who has been survived when she had been treated by this chemera when she was 6, and you can see that she's healthy. So she's the main reason why the FDA approved this drug. And her parents told the court which was responsible for approving the drug, and her parents said that it was very effective, and her daughter was eventually able to make a good life of it. So anyway, effectiveness should be shown in other types of cancers, not only including leukemia. And also, for CAR T cells develop, they also should discover new biomarkers, including CD19. And also, they should need to focus on other types of cells and post-transformer modifications to make their effectiveness increase. So this is my end of my presentation. Thank you.
Okay, thank you, Dongwoo. Questions to Dongwoo?
In the image that you showed in drugs on water, number 8 slide. So I was curious because the name of the drugs has Kim, kind of Korean. So at first I thought that the drug is related to the Korean's body or something, but in the image it said KYM. So is the name is KYM is correct or?
I think I made a typo. Sorry, it's
okay. I see.
Yeah, sorry. Why did I make this mistake? Sorry.
So those drugs does not related to Korean or something?
Yeah, I think so.
Oh, sorry.
Would you show us again the map of Boston? Where is the Harvard you said?
Down over here. It's not shown because I cropped it, but it's like over here. And
I think the Harvard is on the upper side.
Really?
Harvard Square is on the upper side, near to Middle East, that area, or upper and upper. Maybe the south of the river, there may be Harvard Medical School, but main campus of Harvard is
sorry. Yeah, I thought I confused with that. It's actually the Harvard Medical School is over here. So this is the place where many clinical trials are being developed. So the actual Harvard is over here. Sorry.
Okay. CD19 is actually the marker for B cells. So it's a marker for the general B cells. Then this drug, the chemia, can be used only for B cell originated leukemia, maybe?
Yes, they could only be used in B cell leukemia. So in those patients, their B cells are being turned into cancer cells. So the CD19 targeting drugs only are effective in treating those.
Okay. You mentioned that there are two kinds of the drugs that they are working on. They have a different targets.
They're quite I think similar, but also having different aspects of the B cells being damaged. So I think it's different kinds of B cells, but still the same.
Okay, would you go to the slide of the receptor shape? Okay, here. Now, guys, if you look at this, the two structures, normal T cells, the TCL is binding to the peptide, and let's just say the peptide is your antigen. And if you change it into the CAR T, now here the red color is reading as a target domain. The biggest difference between these two, anybody can notice and can tell? Why CAR T should be the better recognizing the target comparing to normal T cells? Let's just say if you introduce this schematic, the antigen receptor, the single chain FV, it may have this original TCL2. You don't need to delete it. Then what can be the difference, or maybe the advantage of this the new receptor may work better than original T cell receptor?
The CAR T cell does not need a peptide between receptor and MHC.
CAR T cell does not need
Peptide between those receptors between CAR T cell and tumor cell.
answer is close, but its focus is a little off. CAR T also needed to recognize antigen, right? That antigen can be maybe peptide, maybe different peptide, but CAR T does not require MHC. Okay, CAR T's receptor is an antibody-like structure. So antibody-like structure do not require the presence of a MHC. You can directly bind to the target. So that's why this peptide, original of the normal cell cases, this peptide cannot be too big. And also it needs to bind, partially bound to MHC, and it can be presented. But if you are other target domain here, other target, it can be huge protein or maybe small peptide just expressed on the cell surface, does not need to have MHC assisted presentation. Okay, so that's why this CAR T has different mode of binding. That's why some difficult target, which is difficult to be expressed by MHC, it can be also a target of this CAR T receptors. So that's another unique other part of this CAR T. So we also mentioned the downstream of the changes, and you said that this is Kimia is a second generation. Could you explain that part again? Yeah, here.
Okay. So so there are 3 generations of CAR T cells that have been developed, and as we heard in the last lecture, the second generation is developed to increase safety, I think, and the third generation was to increase the efficacy. So in the second generation, what Carjun has developed was a second generation CAR T. So,
So what's the difference of the second
signals.
generation from first generation?
It added different receptors, additional receptors. So in here, there are additional signal proteins. So this protein makes, I think this is also in the book Bioscience. So I think if we could take that as a reference, so it has more receptors that it could make it more safe.
I think we covered in the class 2. There's the 3 different the generations. Original one, and also one was a PI3 kinase, the pathways, was helping the survival of the CAR-T, and the other one is a secreting the cytokine the production. So here, if it says a turbocharged, maybe that helps the survival of the cells, maybe. On the right downside, you said these cells are the turbocharged cells and serial killers. So single cell can destroy maybe 100,000 other cancer cells. So second generation, maybe it has a PI3 kinase pathways. It can help self-proliferation or the surviving. And then the different the motif, PLAF2 can also help the function of the cells. So you can add further and further to make this CAR-T stronger and stronger, I think. Okay, guys, any further question to Dongwoo or all the speakers for today? Okay, this week we studied about this the antibody-based drugs and also cell-based drugs. And before that, we looked at the antibody drugs for targeted cancer therapy and small molecule inhibitor for targeted therapy. And before that, we studied for general cancer drugs like Taxol or the DNA attacking molecules. Would you open up the chatting window and type in your most preferred drug style, not the specific name. If you do your research or if you develop your own drug, which drug do you think are the best way? Small molecule second generation, or small molecule third? So far there was no third generation for small molecule. But in principle, if you think it's possible that you can replace those antibody with a small molecule if it can bind to the same binding site, right? So the question is, so we have seen small molecule drug and also antibody drugs and cell therapy, the cell drugs. So if you choose your own research, which one would you like to choose? Type your answer in typing window. Okay, Juan chose antibody drug. Oh, cell therapy. Gene therapy, wow. Everybody gave the answer. 1, 2, 3, 4, 5, 6, 7, huh? Wow, only one small molecule. And gene therapy, antibody is the main. This is a little difficult situation. How do you define Dongwoo, gene therapy? What is a gene therapy?
To alter the DNA sequences. I know it doesn't follows with the selections that you have said, but still, I think it could be effective.
Okay. Okay, Tom, would you take down your file? Now I got a little problem. So I was just thinking, we covered those different the kinds of drugs so far. And for next the week, I was thinking, after studying all of this, my original plan was maybe next Tuesday. No study material for next Tuesday I'm going to give. But instead, I was thinking next week the Sejin Sanbei will present the page display the technique, which is quite useful to know. So there was one plan for Wednesday, the Tuesday. Another plan, I was thinking to give a group discussion or group battle. So if there's some distribution between chemical drugs and biological drugs, and you can support your the preference. That was the original the plan I had. Fortunately, we have one chemical drug, but that's only one. So if Young-hoon should fight against all six, then that's not fair. How should we do this? Young-hoon, what do you say? Or anybody
Actually-
can go ahead.
Actually, I talked about small molecule for biological drug.
Small molecule for biological drug. What is it?
You asked this question about to us, and at the last, small molecule for immunotherapy also can be possible, and I thought that if small molecule for immunotherapy can be developed, it is more beneficial than our antibodies, I think. So in that view, I chose small molecule.
Okay. Okay, then changeable plan. I don't think that we have the even distribution for this, and it may be difficult for Young-hoon to come up with the new things. Then let's do this. Next week, the third day, I had a different plan what to do. Okay, I will the put up different study material for next week. There was some missed part during this class. I will put it up for study. It may take a little time. I didn't prepare for it, so maybe a couple of hours I need to prepare for them and then put it up. So Sejin, could you do this page display presentation next Tuesday?
Of course, yeah.
Okay, then I'm going to teach you about the small molecule or the drug selection, the so-called Lipinski rule for next Tuesday. So change of plan. Okay, any question so far
So
for
we
today's class? Yeah.
should make a presentation as we do by next Wednesday.
No, next week no presentation. So then let me tell you the whole plan. So the last week. So we have 2 weeks from now on. So next Tuesday, I was thinking to do this the small the group battle as a practice of the group work. I thought it can be a little difficult because you cannot talk to each other in face. So you should do this the discussion through the Zoom. It could be also a little challenging. I was worried. So let's cancel this part. But the next week, the material, the very last week, I was thinking, you guys you learned all those techniques and also concepts of drug development and also different type of drugs. So vaccines, even we heard the gene therapy and cell drug or antibody or protein drugs or small molecules. Okay, so the very last week, I will make you call that you prepare your own proposal or the interesting topic study for current coronavirus. So we started with the coronavirus in the beginning. And for next Thursday, I will provide some news articles on the website. Then you will study about it then you do not decide your topic yet. And on Thursday, you are going to discuss, lead by yourself, and then you discuss and to pick up the topics which can be good topic for making the 7 presentation in a format of a conference. Okay, that will be done in the last the week. So 4 talks and 3 talks in 2 days. So that discussion will be done next Thursday online by Zoom. Okay, that discussion. Well, here the Sejin may be most senior. May I ask you, Sejin, to be chairperson to lead that discussion?
And it's my pleasure.
Okay, then I will put up those old newspaper, the news, the articles that's written in Korean, so it will be easy to read. So next Tuesday we will have a class, Sejin's presentation, and also I will teach you some of those small molecule the drug property. And on Thursday, based on those material you read from the newspaper, you just think about the good topics. It can be any aspect of the coronavirus. It can be inspection, screening, drug development, or vaccination, or policy, anything. So which topic you are going to make up, that will be discussed on Thursday. Okay, based on that, you will take one topic after that discussion. And then next week, no presentation by you. But the next week, you will have a presentation. So the deadline will be the weekend of next week. That is the plan. If it is not clear, then you can ask me a question.
So will the conference go with Korean or English?
You decide. Even
It's up to me.
next Thursday discussion. Well, why don't you try in English first? If it's really, really difficult, then maybe you can think. So I will watch your discussion. Sometimes it's easier to do the discussion in English. Then there is no need to think about the tone to junior, tone to senior kind of thing is sometimes a little more complicated rather than just exchange those informations. Okay, so the language sometimes is not bad. It's sometimes a little more convenient. So you may enjoy that part too. So during the whole semester, the main communication is between you and me, and a little questions between you. But this time, I wanted to give you a chance, the full discussion and also organizing those talks for yourself. Okay. Anyway, I will put this the guide onto the website too, so you will not be missed. Okay. Any other question? Okay.
So,
Then if
yeah.
go ahead.
Could you upload the full timeline for those discussion in
Yeah, I will open up
iClass
to that.
or group chat?
Yeah, I will put up that
Thank you.
guideline that on the website too. You can check the website next week material.
Okay, thank you.
Okay. Okay. Thank you guys. See you next week.